Okay, good evening. Let's see, everything looks like it's working. Recording, camera, audio. I'm recording this lecture, I guess you'd call it, tonight for people that might be curious and interested in this topic of neuromelanin and how I came to my understanding of its role in Parkinson's disease. It seems that some of the ideas are potentially very paradigm shifting in medicine, and I had some talk over the weekend or Friday, I guess, with a gentleman who contacted me a few months back about his interest in what neuromelanin was doing in terms of his interest in photobiomodulation. That is, there's been some reports that the use of red light therapy has aided in Alzheimer's disease in particular, but in a lot of interesting things. So this seems like it's potentially very groundbreaking, and it feels like there is very much a shift in the air in science and medicine that's we're on the cusp of that I happen to play a part in, and so it's sort of exciting for me, and I think some people would be interested in what I have to say, and ideally get you thinking about your own understanding of these ideas and what you learned in biochemistry or biology. I'm trying not to get this so it doesn't reflect off my reading glasses. That sort of probably bugs me a lot more than it bugs anybody watching. But so I'm going to try and make a recording, and it'll probably take me maybe 90 minutes or something. I made a slide presentation for the laboratory of Dr. Gary Miller, who's also involved in Parkinson's disease, and he was at Emory at the time when I was trying to get a publication into the literature, and one of the reviewers on the paper that I was trying to get, who I think it was probably Dr. Luigi Zecca, told me that I should reach out and consult with colleagues in the area, and I was at a small undergraduate teaching institution that I didn't really have any colleagues that were doing Parkinson's research, and so I reached out. I did some searching, and Gary Miller's name came up at Emory. He invited me to his lab and was interested in what I had to say, and that gave me the courage to put together this slide presentation, PowerPoint presentation, really. So I'm an old geek, so we used to do slides back in the day. And it lays out sort of the history of my thinking and where I came from and how I came to understand the role of neuromelanin. And so it's sort of a historical perspective. I expect this to be sort of the first recording in potentially a series that lay out the medical and scientific implications of photoabsorbing pigments in human brain. So without further ado, with that brief introduction, I've got a PowerPoint presentation, and I'll just work from that, and I'll probably talk much more than I intend to, but I'm not positive. I think I might be able to draw on these slides as I'm talking. I'm pretty sure you can sort of see me in the corner and hear me talking, and I think I can mark this stuff up, and so I think this is a real good way to present this material. Again, this is now several years old, almost 6 years old, but the ideas that led me to it are quite a bit older than that, and in fact, it's been 300 years since the brains of Parkinson's patients were first chopped up and analyzed, and it was found that the tissue was white relative to their normal counterparts, and that the loss of pigmentation was really the hallmark of Parkinson's disease. And so to me, as a biochemist, not coming from the neuroanatomy perspective. It seemed sort of absurd to me that this thing existed in the core of the brain, and yet the medical community was dismissing it as uninteresting and non-functional. So, excuse me just a second. Edit that out if I can. Hopefully, the booger's hanging on my nose or anything. So this was the title, Neuromelanin is not a spectator. And those of you that know who I am already, that's probably the thing that you associate with me is the title of a paper. It's really a short mini review hypothesis paper that I managed to sneak into the literature that has gained a lot of attention and which drew a gentleman to me recently, which led to some more recent breakthroughs regarding Alzheimer's disease. And so these talks are going to continue as I sort of re-examine everything I know about biochemistry. Okay, next slide, please. Let's see, I'm not sure how to. Oh, I should. Okay, maybe I just didn't click it correctly. Okay, here, this mini map here shows a little bit about me. Just again for some historical perspective and what led me to think about the ideas that I think about and why I came up with the conclusions that I'm going to present over the next several weeks and months and years. I grew up in Seattle, Washington, up there in the left-hand corner. Nice and rainy, not very much sunlight. And I love rain. I love that rainy. I love the trees and the sound of the rain in the trees. And what's in there, Seattle labeled there, is my postdoctoral work. But if you go over there to the right-hand side in Washington State, still up in the upper left of the United States here, when I was getting ready to go to college, a lot of my friends were going to go to the University of Washington, and having gone to high school in Seattle, that, I mean, no offense to them, but it felt a little bit like just an extension of high school, you know, that I was going to end up with a lot of the same friends that I knew in high school and would do a lot of the same things if I went to the University of Washington. And probably more importantly, I didn't want to feel like I had to, you know, go to my parents' house on a regular basis when I was in college. I sort of wanted to move away and be in start a new life, you know, right? When you go off to college, you don't really want mom and dad looking over your shoulder. And 300 miles away, Whitman College was perfect, far enough away without being too far. So that's where I went, Whitman College, liberal arts, New England College, and the liberal, in the New England tradition, I forget what they call it, but definitely challenging, definitely thought-provoking, definitely got a lot of good friends in college, but went back. So I got a chemistry degree at Whitman, biology minor. There was a time that I debated about English, but it seemed like chemistry was more likely to pay the bills. I graduated from Whitman and worked in Seattle for a time at a company that did titrations, essentially. It was a small company that did analytic work, chemical analytic work for Boeing. It was a subsidiary of Boeing, where Boeing had to electroplate or otherwise surface the metal parts for the airplanes. And they do that in these giant baths that contain things like hydrofluoric acid. And so I ended up sort of being a grunt, you know, doing titrations. And I happened to get a position also part-time at the University of Washington as a lab technician. And in short, I realized very quickly that I couldn't do what I wanted to do with a bachelor's degree, and that I sort of needed a PhD. But I spent a year back in Seattle after Whitman, and then applied to the biochemistry program. Now, the thing was, my GPA at Whitman was a 3.0 overall, and I had a 2.8 or 2.7 or something in my major. I gotten a D in biochemistry as an undergraduate. I'd gotten a C and a C plus in organic chemistry. So I was not a, I didn't take immediately to the chemistry, the biochemistry. It took me a long time to really, for this material to sink in. And so that, certainly don't feel bad if you're struggling to understand it. Okay, so the PhD program then, having a 3.0 GPA, 2.7 in my major, there's no way in hell I was getting into a graduate program in biochemistry. I mean, the programs were 3.5 GPA minimum, and it just so happened that Washington State University, this sort of podunk little college that was still a land-grant institution and had some fair good research funding, was just starting a PhD program in biochemistry, and they were doing it by splitting off from the chemistry program. So I applied to the chemistry, essentially PhD program, but I was able to just choose biochemistry for my essentially thesis project. And so I sort of slid in the back door, again, those of you that have struggled with, trying to learn material, it was not an easy path for me by any means. Okay, so, and then I'm getting off track like I will do for the entire time, but take it for what it is, hopefully interesting. At that program, the person that accepted me in their lab was Bruce McFadden, who was studying the fixation of carbon during the Calvin cycle of photosynthesis. We were studying an enzyme called ribulose bisphosphate carboxylase, and that enzyme is active in chloroplasts. And I thought a lot about chloroplasts and thylakoid stacks and chlorophyll and membranes that are associated with chlorophyll. Okay, and so I got my PhD by learning how to manipulate DNA. The thing I was interested in was not photosynthesis or plants at all, but Washington State University being a land-grant institution, that was a primary focus. And so that's how I was able to get into a program that allowed me to manipulate DNA and learn the things that I sort of wanted to learn. So part of the story is back in Seattle when I was in junior high school, we did a little thing and the teachers asked us what we wanted to do when we grew up. And I understood, my dad being a physician, that I wanted to do something in science related to medicine. And then my answer was that I don't know, they haven't sequenced the human genome yet. And so that turns out to be very prescient. I thought when I set out that we were going to learn all the answers to the diseases, the big diseases that we didn't understand, once we sequenced the human genome. Cancer was one of the big ones at the time, for sure, that people were focused on. So I wanted to learn the tools. I wanted to know how to manipulate DNA. And what I did was learn how to mutate the gene that encoded for that enzyme, ribulose bisphosphate carboxylase. So I learned a technique called site-directed mutagenesis, where we predicted that an active site, arginine, in that enzyme was critical for binding phosphate residues of the ribulose bisphosphate substrate. And so I started thinking about phosphate very early on and the bidentate nature. There's a delocalized charge that is on that phosphate group, because it turns out it's critical that arginine is binding it and not something like lysine, because arginine has a delocalized positive charge which matches the delocalized negative charge on the phosphate group. And so that binding pair is a key interaction in a lot of the enzymology. And that was my thesis project, effectively. So got my thesis, my PhD then in biochemistry from Washington State University in 1993. I took a brief postdoctoral position in California. That's not labeled on the map here. That was studying microtubule formation, it turns out, which is also relevant to understanding Alzheimer's disease. But he was interested in centromeres, DNA, and other things. And it was essentially a radiation oncology research laboratory that I worked at for some 3 months or something. And that postdoctoral position was an abject failure. And I went back to Seattle in 1994. I had been a heavy drinker in graduate school, and I decided to go back to Seattle and hang out with mom and dad and go to AA meetings and try and wonder if I'd chosen the wrong career because my first postdoc position flamed out. I won't go into those details. But around about summer '94, I started checking the Seattle newspapers, and at the University of Washington, I saw an advertisement in the medicinal chemistry department. Dr. Alan Reddy was an up-and-coming professor, and he needed somebody that could manipulate genes. He wanted somebody that could do site-directed mutagenesis, and he wanted somebody that could do that on a human drug metabolizing enzyme. You know, this is the same concept I cut my teeth on as a PhD, and I, raised my hand effectively and said I could do that, and he hired me pretty much on the spot and put me to work and had absolute faith in me and could care less about my drinking history. And so I flourished under his guidance in the University of Washington. I learned how to use baculovirus. It wasn't as simple as manipulating small circular plasmid DNA and altering it and taking it in and out of bacteria and E. coli typically, which is the way that simple gene products are made. And even my PhD project involved expressing a plant, a more complex enzyme, but it was still in a bacterial system. You couldn't do that with a lot of the human enzymes. It turns out they need the eukaryotic environment in order to fold properly. We didn't understand that at the time. It's related to the heat shock proteins and chaperonins. But what I did was become very successful at making human enzymes, which made me an expert in pharmacogenetics, because it turns out everybody's got a different subset of allelic variants of a superfamily of enzymes that we call cytochrome P450s. And what they do, one of the things they do is they absorb light at 450 nanometers. If you reduce the iron that's in the heme of that enzyme in the presence of carbon monoxide. And so that's shown in this little diagram here. And so I thought about hemes a lot, and delocalized electrons and the reduction of iron and all the implications of that, and thinking about the superfamily of it. I'll come back to that on the next slide, but just to sort of follow up how I ended up where I am currently and some of the underlying things that happened. I got a grant to study the cytochrome P450s. That grant made me, the University of Washington had to give me a what do you call it? Raise, not a raise, but I mean, I became an assistant professor instead of a postdoc, because I had to be an assistant professor in order to administer the grant, and the university wanted the money from the government that I got by applying for an NIH grant. So they made me assistant professor. I had a grant from the NIH, actually the National Institutes of Environmental Health Sciences, and that made me a hot property for West Virginia University. And so I ended up in West Virginia University, where I had my epiphany regarding metabolism in the brain and its relationship to the enzyme that I was studying. And so I'll go through those details. That is, what enzyme I got the grant on, why I began thinking about neurons and neurochemistry, how I latched onto the idea that melanin was an important molecule in the brain, and how that led me to understanding its role in Parkinson's disease, and now to its relationship to Alzheimer's disease. And so West Virginia University, unfortunately, when my grant money ran out, denied my promotion and tenure. And I essentially came to Georgia with my tail between my legs, and started over again with these ideas in mind that I'd had in West Virginia that I couldn't let go of. I was able, fortunately, to do a few projects with undergraduate students that got me able to publish on neuromelanin and got me some credibility in the field of neurochemistry. And after that, I snuck in my hypothesis paper, which is the thing that's getting all the attention recently and which led to the breakthroughs we and Peter Forehand have had in understanding Alzheimer's disease. Okay, so when I was sitting there as a postdoc in medicinal chemistry, and I was thinking about pharmacogenetics, that is, there are enzymes in the body that react with things that you swallow, drugs in particular, that we would think of being a medicinal chemistry department, and that was our focus. When you take a drug, what happens to it? And I remember asking my dad that, what happens to the aspirin when I swallow it? How does it know where to go? And I don't remember quite his answer at the time, but it has to do with cyclooxygenase. But there's a lot, of course, that we don't understand. And when I was first started out as a postdoc, the enzyme that I was studying was called CYP2C9. And cytochrome P452C9, it turns out, has an allelic variant in the population. We called them, there was two of them at the time we knew of, we called them variant 1 and variant 2. And a lot of the nomenclature has been standardized in the meantime, and the CYP nomenclature, if you can think back to, when I was a student, when these enzymes were just first discovered, and an individual laboratory might discover one and associate it with a certain activity of a metabolism of a certain drug, it might be called, dibrisequin hydroxylase. And so the nomenclature, it was a nightmare to try and figure out that these things were all related. And eventually it was realized that all the CYP enzymes were related to each other. They all had a heme in them. They were all red colored. They all had that absorbance spectra at 450 nanometers. The trouble was, although everybody, it seemed, had an enzyme, one of the variants in that superfamily called 2C9, there were two variants out there, one of them in which an isoleucine at position 359 was replaced with a leucine, and another variant in which an arginine at position 144 had been replaced with a cysteine. And when I was hired as a postdoc, what I got was a plasmid that had the cDNA for this allele on it. In other words, a laboratory that administered warfarin to patients had found an abnormal response in a certain subset of their patients. And they collected the blood from those patients, and they isolated the DNA from the white blood cells in their blood. And they collected the RNA fraction of that, and they made a reverse transcriptase library copy of that transcriptome, which resulted in a cDNA coding copy of the eukaryotic gene where the exons had been removed, and it was effectively just all introns that were in a form that could be expressed conceivably in a bacterial system. So my first job was to mutate the leucine at that position back so it encoded for an isoleucine. And I did that, and that part was easy enough. The harder part was making that enzyme functional in a living system. That is, if you tried to put this on a plasmid in a bacterial system, it doesn't fold up properly, and it needs the membrane environment of the eukaryotic cell. And I didn't understand a lot of those details at the time. I just knew that you had to have a eukaryotic cell to do it. And so I used something called baculovirus. And so that's not entirely relevant to the story, but that's part of the background. More importantly was I was sitting here at this poster for a long time that was next to me that I caught my interest, because this thing on chromosome 22 that was associated with susceptibility to Parkinson's disease and Debrisiquin sensitivity, that turned out to be a related isoform of the cytochrome P450 called 2D6. And so I got hired as a postdoc to work on this CYP2C9. I got a lot of, I don't know what the proper word of fame, if you will. I'm known because, in addition to that, I was successful in expressing a lot of other drug metabolizing enzymes, not just cytochrome P450s, but a group called flavin monoxygenases as well. And a lot of that literature is still being cited slowly over time, and people are looking back and understanding drug metabolism and the genetics of it more. I used that as an opportunity to potentially get funded for myself. That is, my boss went out of town for a couple months. I decided to write a grant proposal on CYP2D6. I locked myself in the garage and started writing my ideas up on susceptibility to Parkinson's disease. So a little close-up view, I mentioned that the alleles of the CYP2C9 gene, turns out that was related to warfarin metabolism. And I think I said already that it was identified because some patients in a warfarin clinic had an abnormal response to warfarin. And it turns out that it was related to a single amino acid change, an isoleucine to leucine at that, which is mind-boggling if you race to think about structure and function as a biochemist like me, because isoleucine and leucine are, you think about them as effectively the same thing. That's the most conservative amino acid substitution you can imagine. And yet that single change effectively destroyed a person's ability to clear warfarin. And so, wow, okay, well, something's going on there that we don't quite understand. But it was a relatively simple system. That is, there were three, known allelic variants. It turns out it's more complicated than that. There's some more rare variants out there. But I got interested in that one that was associated with Parkinson's disease. And so I'll go into that why, how, and why is it, what's it have to do with Parkinson's? Because it's not obvious, and it really has nothing directly related to Parkinson's disease. It just led me to the ideas. What it did have was a lot of identified allelic variants. So this diagram is showing the intron structure of the gene encoding for CYP2D6. And what you're seeing is a review article that was noting the amino acid substitutions associated with changes in the coding frame of the individual introns that made up the coding sequence of this thing. And so I identified some of them here. In one of the alleles, for example, there was a threonine isolute, threonine 107. If you counted up all the amino acids in the chain that made up CYP2D6, the 107th one in the DNA coding sequence was a threonine in the normal gene, and it was an isoleucine in this variant. Then, for example, and there was a serine 486 was changed to threonine, and I can't read the other one, but this was one of the alleles that I chose to alter to see its function in relationship to the normal, what we call the normal allele in the population. And I chose a couple of other alleles, and I set out to do a study analogous to what I had been doing with the warfarin metabolism. But the reason why this gene was interesting to me had to do with its family of substrates that metabolize. I'm looking ahead of my slides. That is, this thing is involved in the metabolism. It turns out some 30% of some all administered drugs are metabolized, at least in part, through CYP2D6. So this enzyme is involved in their clearance. It has something to do with their pharmacokinetics. How long do they stick around in the body? What do they do? Where do they do it? Well, the thing that caught my attention was that not only associated with Parkinson's disease, but everything that's psychoactive. Let me jump ahead one slide here and I'll come back a couple slides. Here's a table showing the substrates for this enzyme. And any of you that are physicians or associated with prescribing drugs, I mean, you'll recognize very quickly these are all psychoactive. I mean, or antiarrhythmic. I mean, these things act in a central CNS fashion, right? These are active in the brain, all of them, right? And that really spoke volumes to me, because what do we think about when we think about drug metabolism? Well, if you went to medical school, you probably think about it as being a liver phenomenon. That is, cytochrome P450s are in the liver. What do they do? When they make hydrophobic drugs more water soluble, so the body can eliminate them in the urine. That's true to a certain extent, but it ignores completely the tissue-specific action of these enzymes and the role that they played in endogenous pathways. That is my interest in because I know the cavemen weren't taking Prozac, and they weren't taking fluoxetine. I mean, they weren't taking ecstasy. I mean, that's what I meant to say. And yet, presumably, they had this enzyme that had evolved, and it was active in the brain, and there was evidence that it was expressed in the brain, and I wanted to know why. And that's where the grant money came from. And so back a little bit here, I also was potentially of the mind that it was a critical enzyme. But if you look, it appears, so this diagram here, I guess I should take a second to explain it. This diagram is showing some of the allelic variants that I just showed in that previous diagram. So the star 1, star 2, star 4. If we go back to that previous diagram, that corresponds with this star 1 thing at the very top, which is the normal allele. The star 2, was it? And star 4. That's the star 2 on here that I sort of circled here. That's this allele. And so it's got 3 nucleotide substitutions, and the star 4 allele, and it turns out that has suballeles on it, A, B, C, and D. It keeps getting more and more complicated every time we discover, sequence more people, and we realize that this is not nearly as simple as we thought. Well, this star 4 allele, if you can see with this term right here in this column, it says activity. It's associated with the activity of that allele. And the normal one is normal. The star 2 allele is decreased. It says decreased activity. And so this was known from pharmacokinetic studies that dobraziquin, typically, if you looked at how fast dobraziquin was cleared in these people, it was slower if you had the star 2 allele. If you were homozygotic carrier for the star 2 allele. Well, if you had 2 copies homozygotic for the star 4 allele, you had no dobraziquin activity. And what we had done at the University of Washington when I was a postdoc, one of the advantages of working there was that there was a human liver bank that, because we were down the hall from University Hospital, which is where I was born, it turns out, and my dad was working in the lab of Earl Davey, who was the guy who discovered the blood clotting cascade. And I was born there. And anyway, down the hall from where I worked as a postdoc, they had an agreement with the hospital to collect the organs of anybody that was an organ donor. And so when accident, typically auto accident victim would come in, the people that I worked with would walk down the hall and work with doctors and cut out the liver and pop it in the freezer that I worked in right across the hall from me. And so we took little cubes of this liver tissue, give it a number, for identification purposes, so it's anonymized, and grind it up and run it on a polyacrylamide gel electrophoresis, which separates proteins by size. And because I had been able to make this enzyme in a baculovirus system, we were able to inject it into a rabbit and generate antibodies against it. And we use that. This is, I think, might be a figure from Mary Payne's paper, the intestinal cytochrome P450 pi, where one of the things we did was compare the status of protein markers for these cytochrome P450s in human livers. And so we had a set of antibodies that recognize the individual isoforms. And so this is a Western blot where after we'd separated all the proteins that are in human liver 118, for example, you can see a sort of a background fuzz, the faint background signal. That's all the proteins. But what we've done is use the antibody against what we know is 2D6, specific in against not any of the other cytochrome P450. So it only recognizes 2D6. Then it picks up the presence of protein in livers that have star 1. That is, homozygotic carriers of the normal allele have this amount of protein as detected by an antibody that binds to it, and then we stain it with the stain in an enzymatic thing that's attached to the antibody. It's called a Western blot. We'll look up the technique, but it's sort of standard. And here are some livers that have homozygotic for the star 2, which has decreased activity. Notice they appear to have an increased amount of protein in them, as if there was a upregulation to try to overcome a decrease in activity on a kinetic basis. And if you look at heterozygotic carriers of these alleles, star 1, star 4, star 2, star 4, they appear to have a normal amount of, apparently normal amount of enzyme in them as detected again by an antibody. But if you're a homozygotic carrier for that star 4 allele, the one that has no activity by diperoxyquin, there's no enzyme. It's not expressed in liver, right? And so apparently it's not critical. It can't be critical if it's not in liver, can it? But this is the liver, and there's evidence that there's a splice variant that it's expressed in the brain. In other words, those same 9 exons in liver do not have to be expressed the same way in other cells, and they're not. They may not be expressed at all. In the brain, it turns out that some of these exons and introns are shuffled or left out entirely. And so the gene product, the thing that's the end of the day when you put the blocks together that are coding parts of it, it's a different enzyme. It's from the same gene, but it's a different enzyme, right? And so this is why we discovered once you sequence the human genome, you didn't have all the answers. You still needed to know the proteome in addition to the genome. And then we thought once we had the proteome, we'd know all the answers. Sorry, I'm here to tell you we still didn't. But we've got some clues now, and hopefully you'll help me pass the word and help understand it. Okay, so back again to the features of that enzyme. It was the one I was hired to do as a postdoc, the warfarin one. It metabolized drugs that were weakly acidic, and so I'm going to skip that part in the interest of time. But there's some interest in Um, the thing that the 2D6 substrates had in common, again, and so I was raised to think about structure, function. Um, what happens if you alter an amino acid in the active site of an enzyme? What does it do? And organic chemistry wise, if you look at this structural motif, what you see, there's an aromatic ring here. There's a linked, in this case, another ring, carbon chain with some nitrogens on it. And this is really the general structural motif for substrates, things that are metabolized by that enzyme I was interested in. They were all psychoactive. Being a good, experimenter in high school, I tried drugs, and I wanted to know what they did, quite honestly. I mean, I'm not making any more, noble than that. Seriously. So it turns out that this motif is also something that resembles dopamine. And so I've got some slides here coming up. Here's again, they're all psychoactive. I mean, with very few exceptions. And you kind of go, well, yeah, sure, they're metabolized in the liver, but what about their metabolism in the brain? If there's a different isoform expressed, I don't care if it's not expressed in the liver. What I want to know is what's happening in the brain. And so that's what I tried to get refunded on. That is, my first grant that took me to West Virginia was doing mutagenesis, making variants of the enzyme and studying how they responded to things like codeine metabolism, dextromethorphan metabolism, because these all fit that same pattern. Notice here, there's a ring here, it's aromatic, it's a little hard to see. There's a couple carbons and there's a nitrogen that has a positive charge on it. And so the common feature of all these substrates, there's about a 7 angstrom distance between the edge of that ring and the positive charge of the nitrogen. And in this case, it appears that there was an aspartic acid side chain amino acid in the active site, which was complementary to the positively charged nitrogen. And so this was very analogous to what I had been doing with the 2C9 cytochrome P452C9. It was also very analogous to what I'd been doing as a graduate student with ribulose bisphosphate carboxylase. That is, here's a side chain of amino acid in the case of Rubisco, ribulose bisphosphate. It was an arginine. It was a delocalized positive charge in the active site, and it bound a phosphate, which is a negatively charged thing. Here it's a negatively charged thing on the enzyme on a side chain of one of the amino acids, and a positively charged free molecule. And by electrostatic interaction, there's a certain attraction that's a large reason why all of these substrates are substrates of 2D6. That is, when you learn biochemistry, you learn early on that there's some enzymes are really specific for their substrates. That is, I mean, they won't metabolize anything except one very specific stereoisomer or one compound. And yet there are these other enzymes, in particular cytochrome P450s, which are really loose. They don't have as much morals. I mean, they're slutty. Anything that comes along looks halfway decent. It's in a skirt, whatever it is, they'll take it. Put a hydroxyl group on it and send them on their merry way. So that has to do with the structure of and how the cleft of the P450 sort of opens up and just sort of lets things sort of come in and be recognized by these structural features. And there's an oxygen, it turns out, bound to an iron atom in a heme, very analogous in many respects to one you might find in a hemoglobin, but certainly different properties. In this case, it oxidizes carbons, and in the case of dextromethorphan, it causes the methyl group here on this hydroxymethyl to go away as methanol. But that's neither here nor there for this story. The next part of this is that this got me thinking about a grant proposal. That is, I had a grant in hand to study pharmacogenetic variants of cytochrome P452D6. One of the things, back to that poster, what did it have to do with Parkinson's disease? Well, this is a whole nother story in and of itself. So maybe let me even just skip ahead here to a slide a few zones ahead and come back. How did I get to be thinking about neurons when this was a drug metabolizing phenomenon that I should be thinking about liver enzymes? Well, again, I mentioned the idea of splice variants. So this maybe it was related to 2D6, but not the same thing actually as in liver. And maybe the stop codon that was in the one in the liver STAR4 variant, in fact, was spliced out, the one in brain, and so the stop codon wouldn't make any difference, right? It'd still be expressed perfectly fine. And so that was part of my thinking, and there was some evidence at the time, and so I was thinking about grant proposals. What could I do? And we set out to try and identify the expression of that splice variant in some brain tissue, and again, more detailed than I want to go into right now. But one of the things that got me thinking about this was way back when I wrote my grant proposal. A gentleman named Neil Castagnoli was on my mind. Neil Castagnoli wrote a letter of recommendation to the NIH in favor of my grant proposal. And Neil Castagnoli was a chemist who, when I first heard him speak, he boggled my mind. I mean, the way he looked at these chemical structures and could talk about them, manipulate them. And I know you're probably doing the same thing listening to me. And I see them as words now instead of individual letters, and it's very hard to pick up that skill until you spend a lot of years in teaching, things like that. And so anyway, I'll try and go through. This is effectively what Neil Castagnoli talked about when Marie and I traveled to France one time, and when I was a postdoc. It's the MPTP story. It's the reason why that poster said susceptibility to Parkinson's disease related to the cytochrome P450 CYP2C9. The story is something like this. Some kids, okay, so those of you that are old enough will remember the designer drug era. That is back in the early 80s, I think maybe late 70s. In California, largely, there's a lot of, the hippies that started learning chemistry and the government was trying to stamp out that sort of, we can't have anybody having fun, can we? We don't want them thinking outside the box, by golly. And so anytime that hippies would come up with a new chemical that got them high, the FDA would make it illegal. And so there was this game, right? They would alter the structure, make something new that got them high that they could distribute, and they wouldn't get arrested for it, and they could make lots of money and be happy again. Nowadays, that's all done in China, and you have to order it through the dark web, but it still happens, and everybody dies from fentanyl overdose because of it. But the public is not yet ready to accept the idea that humans need a certain amount of psychoactive things in their diet all the time, and it can take many forms, but we don't quite understand that yet. So anyway, back to the story though. Meperidine was an FDA-approved analgesic, if I'm remembering correctly. And alpha protein was another one that was related to it. And alpha protein here, if you'll know the difference between them, if you look at meperidine, it actually acts like an opiate analgesic in a similar fashion. It's not quite the tri-ring structure, but it's got some of the features of it. Well, if you look at a parity, it's got an ester on it up here, this C double bond O with an oxygen next to it. And protein is another analgesic related one, and it's got that same ester in the same position, but it's switched. The oxygen and the double bonded oxygen have reversed places. We still call it an ester, but the ester came from a different synthesis pathway. And this one has a methyl group in addition on this six-membered ring that has the nitrogen in it. And so this is relatively stable. Okay, so these kids in California had the bright idea, we're going to make MPP plus. I mean, sorry, MPPP here in this diagram, the third one in the top left here. This is what they set out to make, because it looks like meperidine, it looks like protein. It is neither of them. It almost certainly is going to act as an analgesic, but it's not FDA approved. It's not going to get us in trouble. We're not going to get arrested. So they made this. Unfortunately, this ester, when that methyl group is not on the ring, is not stable. That is, under heat, if you get your reaction too hot, that ester eliminates. And when it eliminates, it does more than that. It ends up leaving a double bond in the ring. And so it's partly now oxidized. And okay, so I think I can erase that stuff. Okay, and so let's try to clear some of that up a little bit. Then what happens then if kids in California inject this stuff into their veins? Well, they didn't get the MPP plus like they thought they got. They wanted this thing. They got this thing. Okay, so what, they probably still got high. It still had the same sort of basic motif, but the ester was gone. It had eliminated, and in its place there was a double bond in this ring. So what happens in the body from there? This is where the enzyme that I had a grant to study comes in, because I was studying this P450 here, CYP2D6 in liver or potentially in brain. I wasn't sure, but I wanted to understand that. If you had functional copies of that cytochrome P450, then the stuff that these kids injected, the MPTP, was converted to something called phenyl tetrahydropyridine. So MPTP is methyl, here's the methyl group, phenyl, here's the phenyl group, and tetrahydropyridine. So pyridine, whoops, let me start over again. Pyridine is this guy. Pyridine is a six-membered ring that has double bonds alternating. It's aromatic. And if you have tetrahydropyridine, then you put hydrogen here, hydrogen here, hydrogen here, and hydrogen here, and you've got 4 hydrogens, and you still have a double bond. I mean, it ends up here, and so I didn't quite count that up for you quite right, but I don't want to fix it right in the moment. You get the idea. Ideally, if you put 4 hydrogens on that, you got pyridine, tetrahydropyridine. And so this is methylphenyl tetrahydropyridine. Here's the tetrahydropyridine, here's the methyl, here's the phenyl. Okay, so MPTP was converted by that enzyme, CYP2D6, to phenyl tetrahydropyridine. In other words, much kind of like the dextromethorphan, it hydroxylated the methyl group. In this case, not a hydroxymethyl, but a nitrogen methyl. And the CH3 group, the methyl group, went away as methanol, effectively. And what you were left with was called phenyl tetrahydropyridine, because the methyl group was no longer there. And this guy is non-toxic, completely non-toxic. And so it was eliminated. Those guys went away on their happy, merry way, getting high. But sadly, 10% of the Caucasians, and that's only the numbers we have for that subset of the population. We don't have numbers for African Americans, for example, or Asians, because the allelic variant percentages differ drastically in different populations. The thing that happened, what if you were a star 4 homozygotic carrier for that cytochrome P450? What apparently was happening was that 10% of those patients, the people that were injecting this stuff directly into their veins, now mind you, MPTP had been worked with in organic chemistry labs for decades without any problems. Absorption through the skin didn't do it. It was direct injection, and apparently the inability to detoxify it, which was the issue. And if you didn't detoxify it, if you didn't have that P450 enzyme, some of it was detoxified or eliminated through the conversion to an N-oxide through one of the flavin monoxygenases. But a big chunk of it, here in this heavy arrow to the right, had, I should say, the pyridine, the tetrahydropyridine, reoxidized back to the pyridine ring. In other words, 4 hydrogens were removed from it. And so it was no longer, oh crap, that suddenly strikes me why that occurs, but I can't get on that right at the moment. I got to rethink everything I know in light of what we thought about over the weekend. So, well, when it becomes pyridine again, then this is the new compound, MPP plus, methyl phenyl pyridinium ion. If it's got the substituent on that pyridine ring, it's got a positive charge. And so, bingo, this looks a lot like those substrates for CYP2D6, right? It's got that same aromatic ring, a couple of carbons, and then a positively charged nitrogen. Well, it turns out that this compound, MPP plus, is the thing that causes Parkinson's disease. And so the kids in California that lacked, the 10% of them that lacked the P450 enzyme to detoxify it, converted it instead through an enzyme that's very prominent in the brain called monoamine oxidase B. If you've ever taken any, you looked at the back of any pills you take over the counter, a lot of them say, don't take this if you're taking MAO-B inhibitors. MAO-B inhibitors were some of the earliest antidepressants, the tricyclic antidepressants, which essentially inhibited the destruction of dopamine by glial cells and caused an increase in local concentrations of dopamine. Well, MAO-B, because it's high concentrations in the brain to convert dopamine to something that isn't quite as toxic in glial cells, it turns MPTP that these kids were injecting into MPP plus, which disrupts mitochondrial electron transport. And so what was absolutely known is that this guy caused mitochondria to stop being able to make ATP. And if they couldn't make ATP, then things started shutting down. We didn't understand that process, and suddenly I do over the last few days. It's going to take me a long time to bring this out and into the literature, but this thing, if you get it into any dopaminergic neuron, and it does that because it looks enough like dopamine, it has that same sort of structure that all of the 2D6 substrates have and that dopamine shares, which I haven't even shown dopamine structure yet. But it apparently was concentrated into neurons that utilize dopamine because it looks like dopamine. And this was really intriguing to me. That is, this thing, MPP plus, looks apparently enough like dopamine that if you inject MPTP into animals, mice, for example, rats, then enough of it gets converted to MPP plus and is taken up into dopaminergic neurons that it causes Parkinson's symptoms. And so this is the typical animal model for trying to understand Parkinson's disease. But the thing that that all of the Parkinson's studies are missing is that, yes, this is all true, that the MPP plus or the MPTP, I'm not entirely sure which one's taken up into the neuron first and which one's taken up into the nearby glial cells. The thing that they're missing is that it's particularly toxic to cells that have accumulated melanin. Okay, and so here I've been thinking all this time about cytochrome P4, this P450 right here. I wanted to know its role in Parkinson's disease, and its role is not directly related to Parkinson's disease. It turns out that if you happen to be injecting something that you're trying to get high on, and if you have that enzyme, then you don't get Parkinson's disease. So it wasn't protecting from Parkinson's disease. It was protecting against chemical insult that would otherwise cause Parkinson's disease. But that's still relevant, very highly relevant. That is, in this particular case, this thing looks enough like dopamine that it causes problems for dopaminergic neurons. And one of the problems it causes is kills the mitochondrial activity. And that creates a situation where the cell goes into apoptosis. And in this case, it turns out that starts releasing melanin that forms in neurons that use dopamine. And so I have to switch to a new figure, but this is what I was thinking about at the time. And what started me thinking about a lot of all this was Neil Castagnoli and his story, the MPTP story, how cytochrome P452D6 was involved in detoxifying something that otherwise caused Parkinson's disease. And there's more to the story, and obviously I could go on and on about it, but this is already getting out of hand. I need to back up. I skipped a couple of slides and I don't know how relevant they are, but this is the one that where I had my epiphany. That is, this was the figure I drew, and I was trying to write a grant proposal because my promotion at West Virginia University was at stake. One of the things I knew, and so this was incorporating those ideas. And so let me show first the MPTP angle. And so what I've sketched here is a synapse of a neuron. And so if you've studied any neuronal physiology, this would be an axon coming down here. Here's the junction between two neurons. We call it the synapse. And I tried to illustrate some of the circular closed lipid vesicles that dopamine accumulates in, and that a dopaminergic neuron releases in response to an electrical signal traveling down this neuron. So what I knew was that there was potentially this CYP2D6 in there, that the enzyme that I had a grant to study, I knew it could potentially convert MPTP to PTP, and it was involved in its detoxification. And so that was illustrated here. I know that there are a couple of different forms of monoamine oxidase. There's a monoamine oxidase A in neurons, and there's a monoamine oxidase B in glial cells. And so this is illustrating a glial cell. It's something we associate with an immune response. It's also associated with stabilizing the synapse once it forms. In this case, I'm illustrating the idea that MPTP, the stuff injected by those kids in California, was being converted to that MPDP plus and then to the MPP plus, and that one way, shape, or another, these things resemble dopamine. And so they enter the dopaminergic neuron, probably with large part of assistance through monoamine transporters, which are fairly nonspecific. They take dopamine, they take tyramine, which is essentially the amino acid tyrosine, which it's lost its carboxylic acid. And it turns out if you eat a lot of aged meats and cheeses, they have the proteins have hydrolyzed and a lot of the tyrosine in them. So let me sketch that out real quick. I'll try and draw it over here. Here's the amino acid tyrosine. And we would draw the aromatic ring like this. And tyrosine has carboxylic acid here and an amine here. And so here's the alpha carbon. Here's the amine attached to it. Here's the carboxylic attached to it. And so this is an amino acid by definition. In this case, the side chain is this thing here, where it's a CH2, an aromatic ring, and an OH group. These are often target of phosphorylations in proteins, which turns out to be very relevant to some of the more recent hypotheses on how the phosphate is affecting the shells of both vesicles and water that form around phosphorylated compounds and other things that assist in electron delocalization. A lot of these concepts are very advanced and are going to take some explaining and convincing for people. So the thing that happens if you have proteins in like sausage, cheese, it sits around years, decades sometimes, in a cave somewhere. Well, what happens? These carboxylic acid and the amine would normally be tied up in peptide bonds, but in a chain of other amino acids. But over time, when tissue stops living and the peptide bonds begin to break by the addition of water across them, then you end up with free tyrosine. And the thing that happens with free tyrosine over time is the carboxylic acid eliminates. And it can do that because there's a positively charged group here, essentially stabilizing the elimination of the CO2, which necessarily leaves the negative charge behind. So what tyramine is the thing that's left. If you look at this thing after you've taken away a carboxylic acid, that's tyramine. It's essentially the amino acid tyrosine, which is essentially one of the chunks of proteins that's sat around for a long time. And one of the things that happens that I knew and read about was that if you eat tyramine and you're taking MAOB inhibitors, well, MAOB, monoamine oxidase B, would normally take the tyramine that it encountered in the glial cells and eliminate it, convert it essentially into something else that I'm not going to try and show the structure. It deaminates it, and it's no longer an amine, and it's no longer a neurotransmitter or psychoactive or anything of the sort because it doesn't have that positive charge on anymore. But if you are taking MAOB inhibitors, you can't do that. And if you eat aged meats and cheeses, the tyramine instead causes something called the cheese effect, where you make too much dopamine. And one of my hypotheses was that it was that enzyme I was studying, CYP2D6, in the brain, or this may have been in the literature already. I'm kind of getting some of it mixed up. That 2D6 was responsible for the cheese effect, that a hypertensive crisis that happened if you ate aged meats and cheeses and you were taking MAOB inhibitors. Okay, so I knew there was 2D6 in the brain, right? I knew that cytochrome P450 that I studied was potentially involved in making dopamine. But I also thought it can't be essential if it's missing in 10% of Caucasians. But is it the same isoform? There was a question whether it was the same splice variant or whether there was something completely different in the brain that could do one of the activities but not the other one, and that's entirely possible. And I'm not specifying which activities, but okay. And so one of the things that happens when you're metabolizing dopamine is well, I'm going to have to maybe go to a whiteboard here and come back to this thing. To show you, no, maybe I can do it on the next slide. Or I can show it here. Yeah, okay, there's a couple of slides coming up here. Okay, so dopamine, well, I can almost do it right here because tyramine, in order to become dopamine, if you just put a hydroxyl group here, which is what the enzyme that I studied did, it's dopamine. And so this is dopamine right here. It's tyramine that's been hydroxylated here. And dopamine is an example of something called a catechol. And this is an organic chemistry thing that I learned about because Dr. Todd was merciless in trying to get us to understand organic chemistry. That is, these things become quinones. They oxidize. This is a redox process. And dopamine is a catechol. It's a catechol with an amine on it. So we call it a catecholamine, and a catecholamine is typical of a neurotransmitter. And so here's your catechol part, and here's the amine part, 2 carbons and an amine. That's dopamine. And dopamine is toxic to cells because it does this. This is stressful. This pressure, redox pressure, is stressful on a cell. And so the glial cells, that's why they have MALB. One of the big reasons they have monoamine oxidase B is because they're not dopaminergic neurons. And any dopamine that is released by these dopaminergic neurons would otherwise destroy them. And so they have MALB. And that's why MALB inhibitors increase the local concentrations of dopamine. You know, when the pieces start to fit, it's sort of, you start to laugh at yourself because they were all in front of you, but you don't see them until the puzzle picture emerges. So in the dopaminergic neuron, what stops them from doing this? What stops them from the redox? And in the case of dopaminergic neurons, what happens if they do? Well, it turns out that if they do this in the neuron, they make melanin, the pigment, the same stuff in your hair and your skin. You know what makes my freckles? When I was a kid, I was looking at them going, you know, why in the heck do I have freckles, and what are they, and why does that make me different than other people? It's melanin. Why do bananas get brown spots on them? It's melanin. The cell structure breaks down in the banana peel, that it releases dopamine-related compounds, catechols. The catechols react with themselves and polymerize, and the polymerization product is something we call melanin. Okay, so I'm like, okay, wait, you're telling me that there's the pigment melanin in dopaminergic neurons. This was my epiphany moment. I'm going, wait. And not only that, but MPTP plus is particularly toxic to cells that have accumulated melanin. And those cells occur in the substantia nigra that is deep in the brain of adult humans. And so I wanted to know nobody told me about melanin. What the hell is that doing in the brain? What is melanin? And so I'm going to move from this slide. Yeah, okay, here, I've got some slides coming up. I do have been able to illustrate. I'm going to do a lot of jumping back and forth because my thoughts aren't matching the slide presentation anymore. Okay, so a little bit about Parkinson's disease, maybe, and the MPTP story, and thinking about what's going on in the brain. And then we'll come back to the melanin part, the dopaminergic neurons. Okay, so here's a little bit of neuroanatomy, just showing where in the brain we're talking about that this was happening, that cells had a lot of dopamine in them, were creating enough to darken the cell and make it appear black under the microscope if you took tissue slices of it. That was called the substantia nigra. And what was known 300 years ago was that if you took a slice, took a dead person who died from Parkinson's, and you sliced right through here, that it was white. You didn't see the typical black associated regions of a normal adult brain. And so this briefly sketches out what dopaminergic neurons are doing in the brain. That is, in this region, in the substantia nigra, which is the thing that I focused on most, because this is where the pigment is lost in Parkinson's patients. That is, patients that have Parkinson's disease. The earliest thing that was recognized was that this part of the brain that would normally be black was white. And what we're showing here, this diagram is showing, is in blue, that these are the cells that are releasing dopamine to begin dopaminergic pathways that project into the striatum and the frontal cortex and all the other regions in the brain. And so essentially these are muscle movement sort of memory pathways when you release dopamine into these regions. And I'll maybe try to make more sense of that. But I'm not a neuroanatomist, I'm a biochemist. I came at this from a chemistry perspective. And so a lot of this was mumbo jumbo to me at the time. But what struck me was, here's the brainstem. I mean, right, here's the spinal cord. And where is this thing in relation to evolutionary history? It was the first thing. It was the nucleus of the brain. Yeah. Everything else spanned out from that, right? Think about how this thing began. And so that really struck me, that the region very central to the core apparently was making melanin. And as a biochemist, I said, if you're making a polymer of dopamine and it's accumulating and being retained in the brain, it's functional. There's no way in hell, that I said to myself, that the brain keeps garbage around, which is the prevailing view in medicine still today, which is frustrating. Because when I was a postdoc and I was trying to express cytochrome P450s in bacteria, when I made a mutation in the gene that encoded for something that caused the protein to misfold, that misfolded protein was chewed up and spit out of those cells faster than you could say garbage. And yes, sometimes you get tangles of proteins and inclusion bodies, and that was a well-known phenomenon. But the thing that struck me was the body doesn't keep garbage around on a regular basis. And yet here I was looking at something that the vast majority of people were apparently convinced was non-functional. And here it was at the base of the brainstem. And as a biochemist, I said to myself, bullshit, that's functional. If it's there, it has a structure. A structure has a function. The function is not garbage patch, stuff that the brain couldn't get rid of. I rejected that idea out of hand. And so I immediately set out asking everybody I could think of, all the neuroscientists, all the physicians at West Virginia University, School of Medicine. I asked them, what is this stuff doing to the brain? And nobody could tell me what it was doing. When a neurophysician, neurosurgeon told me one of the things, he said, I don't know, but it makes me think about this green glowing stuff I find when I take brains out of the skull. And I was like, what? What are you talking about? And he said, yeah, when I isolate brain from the skull, there's a layer of green glowing UV sensitive tissue on it, light sensitive tissue on it. And he didn't know what it was. And then it turns out I think that's probably something we call lipofuscin, which again is highly related to my story. That is, it's a pi conjugated system that exists in the lipid fraction of the brain. Well, it's going to take me a long time. Some of you might be able to follow that line of thought, but most people are going to struggle until I really explain a little bit better some of the concepts behind it, which is going to take a bit. Okay, so but back to this idea. Parkinson's patients, it was white. Normal patient, black. And the very seed regions, if you will, of the brain when it first started to develop. And they were trying to tell me, after knowing this for 300 years, that pigment didn't do anything. That's how I started laughing at it. I go, well, you gotta be kidding me. It's melanin. If we already know it does stuff. I just want to know what it did in the brain. I mean, that was just nonsense. Okay, so I think let me just scroll back here. One slide. And so just sort of putting this back into context and again, and then we're going to move away from the cytochrome P450 story pretty soon altogether, because it's actually not relevant, but it got me to the ideas. That is, 2D6 is in the brain. It's a cytochrome P450. It may be part of dopamine synthetic pathways. This was a figure from a review article in a book chapter. I think, no, it was a big review article on all of the human cytochrome P450s, which led me to some ideas about really their central role in evolution. These are, yes, the enzymes metabolize drugs, but to call them drug metabolizing enzymes is really simple-minded. They existed in the body during evolution, and they influence normal endogenous pathways. And the drugs that people take happen to influence the pathways that are normal. And so we could call it drug metabolism. Yes, and in many cases, the enzymes are upregulated in response to a drug. But that doesn't really tell you what the normal function of the enzymes is in the absence of the drugs that we're swallowing. And it isn't, they're not just expressed all the time, just in case the cavemen started taking ecstasy, I guarantee you. So here are some of the pathways, and again, it looks really complex, and it is, but the take-home message is that too much dopamine in the brain leads to the formation of melanin. And so I've got some more on the chemistry of that here that I think will help make some sense of that a little bit. So go to the literature, think, okay, melanin, yes. How is it made in the skin? And how is it released into the hair? Which is, that'll send you down a rabbit hole. Try and tell me how the melanin gets into the hair. Well, if you look at what's in the literature about how it's made, it's an enzyme-dependent process. That is, tyrosine, the amino acid tyrosine up here. Let's see, whoops. Oh, how come I can't draw? Oh, here it is, okay. Tyrosine is converted through the action of something called tyrosinase to the quinone form of the amino acid form of dopamine. We call it dopaquinone. And this thing is on the pathway to something where the dopamine polymerizes. And there's a couple of branch points here. That is the tyrosine that is converted to dopaquinone through the enzyme tyrosinase and the interaction of oxygen. If there is the amino acid cysteine present, okay, so what's cysteine? Well, if you've taken biochemistry, then you'll think about, okay, an amino acid. It's got the same alpha carbon. It's got an amine on it that's positively charged. It's going to have a carboxyl group on it that's negatively charged, typically under aqueous conditions. There's going to be a hydrogen on it, and there's going to be another substituent. And in this case, the substituent is a thiol, cysteine. And this is important because thiols form disulfides, and that's a redox process. And so what? Well, cysteine, the thiol form of the amino acid, is present at high concentrations in the brain. And it's there, almost certainly. It's one of its major functions is buffering the redox potential of neurons. In other words, if probably many of you are familiar with the idea of pH. That is the negative log of the hydrogen ion concentration in a cell. And you understand that the pH is critical to maintaining function. What you're not as comfortable thinking about, and probably never even thought about, is what's the flip side of that coin, right? Well, it's the p electron, right? I mean, it's the electron concentration, the redox potential of the cell, effectively. They're sort of inversely related to some extent. It's not quite that simple, I don't think. But what's true is the electrons have to be buffered in the cell just as much as the protons do. They have to be maintained at whatever the equivalent is of pH 7.4. But there's compartments that have different pHs and different redox potentials and vastly different properties because of it. In the brain, that redox essentially maintains dopamine so that it doesn't oxidize to a quinone. And if it does, it's there to essentially detoxify it, in a sense, by reacting with it. And so what we see here is cysteine reacting with this quinone thing, forming the intermediates that then start to grow. And it's hard to picture this, but it's a polymerization process. That is, more monomers of dopamine and more cysteine incorporates into a polymer that we call melanin. In this case, it's the red stuff, pheomelanin. This is what I've got in my hair. Pheomelanin is red, and most of the freckles in my skin have pheomelanin in them. Apparently, I've got a lot of cysteine relative to dopamine. And another way of saying I'm dopamine deficient. And anybody that's ADHD go, surprise, surprise, really? Imagine that. Well, it's more complicated than that, but that's the essence of it. And people that don't have cysteine, or in other words, more dopamine relative to cysteine, they make eumelanin. That is, the polymer of melanin still grows in the presence of excess dopamine. In this case, we're showing, here's L-DOPA, which is the drug they give Parkinson's patients. Because L-DOPA is, for one thing, it's decarboxylated and it becomes dopamine, and that happens immediately when it enters a dopaminergic neuron. And so this contributes to dopamine in a patient that isn't able to take up dopamine otherwise into their dopaminergic neurons. But if you give a lot of that dopamine to that patient, it also oxidizes to the quinone and starts this process of polymerization into melanins. And when there's not cysteine around, what you get is called eumelanin. And again, this is the process in skin in which a number of enzymes have been identified that contribute to that process. And if you studied biology or chemistry like me, then the thing that would strike you is that everything that you're taught has an enzyme associated with it. Yeah. But not in the brain. That is, back to this neuron here. What happens in this neuron if you are firing very rapidly? Okay, so back to some neuronal physiology. What happens in a firing neuron? What does that imply? Well, what happens is that there is a charge. The way it's pictured in a textbook would be that there's a depolarization of the membrane here. That is, let's see if I use this. It's red maybe now. There are sodium ions and potassium ions that are being pumped in and out, or pumped out of these neurons, and they're trying to flow back into the neuron because this is a minus 70 millivolt potential inside. If you read it from an electrode of voltage potential, the pressure of electrons. And so there's an electrical charge essentially that travels down this membrane that we think of as being a signal. That is, if something happens to this neuron somewhere, could be feet away, some of these axons extend a great distance through the body, then that signal depolarizes the membrane, following down along the length of that membrane. When it gets to a junction here, a synapse, you can think about this as an electrical capacitor, essentially a capacitive junction, where in order for the electrical signal to cross this gap, it would have to spark across it effectively. And so if you increase the voltage difference across it, or you change the resistance through a variable capacitor, then you create a point where there's enough voltage to jump the gap effectively, spark across it. And for a neuron, what that means is you release dopamine from these vesicles into the region in between the neurons. And that's essentially thought to be the thing that causes the spark to jump the gap and allow the signal to transmit down to the next cell. And so this is critical then to how neurons communicate. They release dopamine, the electrical signal jumps that gap, it continues on to the next neuron. It also helps explain why positively charged amines are drawn from the gut and other places into neurons because of that internal minus 70 millivolt charge. That's again attained through maintaining an ion balance in these neurons. And that story there's so much more to that. I wish I could have time to go in right now, but this is really just meant to be this introductory lecture again, and what led me to some of these ideas. So then in that neuron, apparently, this process is occurring where we're getting melanin. But the thing that was key, the thing that really stood out to me about that was there was no enzyme that is in that synapse. Apparently, what happens if dopamine is in the cytoplasm and not in a vesicle, it becomes melanin. And this was shown effectively in a couple of different ways. One of them essentially by disrupting vesicular monoamine transporters. And this is where Gary Miller's lab comes in. They were doing transgenic mouse studies where they were manipulating the transporters that essentially package dopamine into vesicles in the synapse of a dopaminergic neuron. And what him and a group, actually David Salter's group up in New York years ago, had showed that if you somehow increased the cytoplasmic concentrations of dopamine by either increasing uptake through dopaminergic transporters or decreasing its intake into vesicles, that you polymerized it into melanin. And what was true about that was that it happened without the necessity of any enzymes. It was spontaneous. It was chemistry. There was no catalyst, no enzyme catalyst. And I'd never heard of such a thing, because what this implied to me was that if the melanin was central to Parkinson's disease, and that MPP plus was toxic to dopaminergic neurons because it was bound in the melanin and it disrupted mitochondrial function, but I got to get to that story still. There is some evidence that when you accumulated melanin in an older dopaminergic neuron, you didn't synthesize dopamine in these neurons anymore. And so I got to go explain more of this. But again, that's a critical idea, that understanding that if this melanin is central to the development of Parkinson's disease, which it seems to be, and it is formed spontaneously, if that melanin has a function, it would not have a gene associated with it, right? And so only 7% of PD patients have an associated gene defect. 93% of Parkinson's patients are idiopathic, unknown cause. And after all this time of sequencing the human genome, you're going to tell me we haven't found the gene for it yet? Well, maybe there is no gene for the root cause of Parkinson's disease. This was my light bulb moment. What if this melanin polymer is functional in dopaminergic neurons? And if you disrupt that function, you get Parkinson's disease. Doesn't that explain an awful lot of things? So, thinking some more about polymers of dopamine, here's what it would sort of look like if you look at eumelanin, the thing without cysteine incorporated, and pheomelanin, the form with cysteine incorporated. It looks like a mess, for one thing, if you're not used to looking at complex chemical structures. One of the things that stands out to me is all of the double bonds in it. That is, all of these things. And all of the carbons that we would consider to be in the sp2 hybridized state that are everywhere in these structures. And that's, again, one of these higher concepts that most people vastly underappreciate. They do not recognize the significance of that. But it's really important. That is, this is what we call a pi conjugated system. And if you're an organic chemist and you study NMR, then one of the simplest pi conjugated systems is benzene, 6-membered carbon ring that has alternating double bonds. And what we say in that ring is that there are really 3 electron groups around each carbon in a what we call a trigonal planar arrangement, 120 degrees angles between the bonds on those carbons. And if you picture the double bonds as an extra pair of electrons between those carbons, it's a little bit of an illusion. And so let me switch colors here and see if I can help illustrate a little bit what's happening in a benzene ring. in this benzene ring, there are carbons at each of these positions. So there's 6 carbons, and there's 1 hydrogen at each of these positions. And the single bond between them is something we'd call a sigma bond. And the single bond between carbon atoms, again, is a sigma bond. In other words, there's a pair of electrons in each of these lines that is tethering the positively charged nucleus to each other. And in this case, they're arranged at 120-degree angles. They're all in the same plane. They're all flat in the plane of the board here. But if you look at the orbital diagram, if you were taught this in freshman chemistry for carbon, you'd see, well, there's a 1s orbital, there's a 2s orbital, and there's the 2p orbitals. And so what we're saying is that in a carbon compound that has sp2 hybridized carbon atoms, that there are 3 electron groups around each carbon arranged in that trigonal planar arrangement. And the way we think about that is that the s orbital in the second shell and 2 of the p orbitals hybridized to create something we would call sp2 orbitals. And there's 3 of them around each of these carbon atoms, and that's what the single lines represent. But there's a leftover p orbital that's not hybridized. How does that fit into this diagram? Well, what we would do is illustrate that as out or behind the plane of the paper. In this case, here's the p orbital that's got 2 lobes on it going in and out of the plane of the paper. And when you put the single electron that's part of the double bonds in those orbitals, what you'll quickly see is, number 1, they're not in the same plane as the atoms. And number 2, it doesn't matter whether you put a line between these 2 carbons or these 2 carbons. So a lot of times we draw these with a circle here because we know that those electrons are not tied between these 2 carbon atoms like the other 2 are. That's something called delocalization. That is, imagine if you take a photon of light and you excite an electron that's in this pi bond. It turns out what happens is that you've created an electron that's excited everywhere in that molecule at the same time, because the electron isn't confined to any one place. An electron is a wave, after all. It's not a particle anyway at this point. And so this means that in this thing, there is a flow of electrons. And when there is a flow of electrons, there is a magnetic field associated with it. And so we know pi conjugated systems have a magnetic field associated. You know that because when you look at your NMR spectrum, where does benzene occur? It's shifted downfield, right? It's way out here at 7 because of the magnetic field induced by the delocalized electrons. Yeah? Now imagine that on steroids in a melanin molecule. Okay, that's what a pi conjugated system is in the case of these pigments that we're going to be talking about. Okay, so here's the figure from the Sulzer paper. And briefly showing that if you overexpress vesicular monoamine transporter, in other words, in those vesicles, you cause dopamine to be taken up faster than it would otherwise. You limit the formation of melanin. This is that evidence I was saying that in the cytoplasm, excess dopamine polymerizes, becomes melanin. This was the evidence for it. And some other evidence involving N-acetylcysteine, L-DOPA concentrations in these cultured neurons that were overexpressing vesicular monoamine transporter. And I won't go into find details of that experiment, but if you go back, that's one of the key findings that made me say, aha, because this convinced me that melanin in the brain was formed non-enzymatically, and I was convinced at that point that it had function. And Luigi Zecca's name was all over these papers, and David Seltzer's name now is on my radar. So here's some more, just again, not again, but not just dopamine. I mean, this is something that norepinephrine is another catecholamine. Adrenaline, which is epinephrine, is a catecholamine. All of these related compounds, presumably, are subject to the same redox process and could be included into these polymers. And this may very well be part of the reason why we haven't understood them for so long. They're not necessarily regular ordered arrays. What they are is pi conjugated systems. And they have certain functions. And apparently those certain functions are critical to adult living things, humans anyway. One of the things they do is they bind things. We already knew they bound MPP+. Here's some literature. A lot of literature I found was in plants, but this was relating haloperidol, which was known to cause Parkinson's disease, and has a structure very similar to MPP+. And I was looking for then the structure-function relationship. That is, could I predict things that were going to cause Parkinson's disease if I understood that anything that disrupted the function of the melanins was going to do it? And it wasn't necessarily related to mitochondrial dysfunction in those cells, but disruption of the melanin function. And nobody could tell me what melanin was doing there. Everybody said it was garbage. And I said, hogwash. We knew the melanin bound stuff. It binds things like this. Here's MPP+, the thing that kids in California discovered for us. Here's haloperidol. And another one is paraquat, it turns out. It has the same structure. And paraquat, you might remember from the days when the FDA was spraying it on crops and pot crops in Mexico, and kids in the United States were coming down with Parkinson's disease. And so the FDA stopped that paraquat spraying program, thankfully. So here's from another paper, this fluoride paper, illustrating the same ideas I've been talking about, but including now some of the ideas, MPTP conversion. But one of the other primary things that's used to create model systems for Parkinson's disease, that is, once people started figuring out that if you kill the dopaminergic neuron, you cause Parkinson's disease. And so they were doing that in rats and mice and all over the place, thinking that they were studying Parkinson's disease. But there's no melanin in the brains of those animals. And so they might have been killing them by adding MPTP, but they weren't doing it in the same way that it disrupts humans. So all of that effort, almost all of it, has been a complete waste, which is really, obviously a lot of people have a lot of money and time invested in it, and they're not going to let go of their ideas anytime soon or easily. But that's what's happening. And one of the other clues here is that something called 6-hydroxydopamine, right? Here's dopamine. It's got the 2 hydroxyl groups here. It's on an aromatic ring. And so that is susceptible to oxidizing to the quinone In this case, here's an amine on it, which is the thing we associate with dopamine. Got the positive charge, but 6-hydroxydopamine has an extra hydroxyl group right here. And the thing that enables is that this thing can form not only paraquinones, not only metaquinones, but paraquinones. If you're an organic chemist, you'll see immediately what I'm talking about. That is, 6-hydroxydopamine can do this instead of this. And tell me that isn't going to be incorporated into a melanin polymer and not be normal. Well, it's definitely going to oxidize easier than dopamine does because it's got 3 hydroxyl groups on the same aromatic ring. So for a lot of reasons, this thing stands out as being, it's going to bind to neuromelanin, and it's going to react with the neuromelanin, and it's going to create a non-functional form of it. And that, to me, explains how 6-hydroxydopamine causes Parkinson's disease. It's sort of an obvious thing when you think about it and see it that way. But in the literature, what they highlight again is that it's disrupting mitochondrial function, and in this case, looking at 6-hydroxydopamine and potentially MPP plus is interfering with mitochondria to release reactive oxygen species, peroxides, things like that, that were disrupting the energy production in the cell and causing neuronal death. And so, yeah, all that is true, but it ignores the melanin part of it. So, skipping a couple figures here. What did that polymer look like? Here were some of the model structures of what melanin looks like. And every time I see these, I have another aha moment. In this form, I see the pi conjugated system. Yeah, but that doesn't really tell me a whole lot. When I look at this and I see the blue atoms here labeled, blue, where's my blue? Here, nitrogen. These are nitrogen atoms. And notice in these models, they're arranged in a nice, neat little square. And if you were an iron atom, where would you go? Well, if I was a bet man, I'd go right there, in analogy to a heme group. A heme group is essentially, well, you take iron and you put nitrogens around it, and the nitrogens all have 11 pairs of electrons, which are essentially coordinate covalent binding to the positively charged metal. And there's a bunch of window dressing around it. In this case, 5-membered rings that are attached by bridge heads and have alternating double bonds, which makes it a pi conjugated system. So I'm not going to try and sketch all that, but that's what heme is. And that's what melanin is. It looks like a polyheme. And so this is, again, I'm going, you're kidding me that most of the world thinks that this is garbage and the substantia nigra of the brain. I can get, no. Look at this stuff. And here's some of the other models of it. Look at these sheets here. The nitrogen atoms arranged in this polyheme sort of form. And then they potentially stack. So here I'm picturing, okay, one of the things that neuromelanins do is they bind organic compounds. Well, they sure shit do. The iron is sitting there ready to take a lone pair of electrons, just like the oxygen in the heme of hemoglobin. And you've also got this nice aromatic ring structure, which is perfect for something we'd call pi stacking, which is, you might think about it as 2 aromatic rings parallel to each other. It turns out actually that's not stable. It turns out they're stable when they're perpendicular to each other. And that's something that I learned with Jeff, I think on his last name. He was also a postdoc at the University of Washington in medicinal chemistry when I was there. And we showed that one of the mutations in one of the cytochrome P450s, that's a phenylalanine or tyrosine, the phenylalanine, I think it was, that we did the crystal structure of it. We purified it because I was able to make large quantities. And my boss, Alan Reddy, was the man when it came to purifying these enzymes out of the insect cell culture that I was making them in. And so Jeff Jones made the crystal structure, solved the crystal structure of that enzyme that I made, and showed that it was a pi stacking arrangement in a perpendicular, not parallel fashion. Anyway, again, this seems like a perfect place to harbor things that, and one of the other things we know, it turns out, when you read the literature and you look at where the melanin is in a neuron, it's packaged up not only in a membrane, but in a double membrane. And if you add chromium to tissue that has been sliced open, and you add chromium salt to it, the chromium is reduced to chromium metal. And in other words, this region of the brain stains, melanin stain with chromium. And they stain because they add electrons to chromium, and they add electrons because dopamine polymerizes and releases electrons. In other words, there is a reducing environment, we'd say, something that has the ability to contribute electrons in that granule of melanin that forms in the brain. And if I was a dopamine molecule and I wanted to stay in the form of having hydroxyl groups instead of oxidizing to the quinone, this would be the perfect place for it. In other words, in the reducing environment, any free dopamine that's there would be shifted to the equilibrium of the free hydroxyl form. And so, yeah, it's thought that in a dopaminergic neuron, you have these granules, the vesicles, where dopamine is stabilized with antioxidants to stop it from polymerizing to become melanin, presumably, and to be used as a neurotransmitter to release into the synapse. And if it's in the cytoplasm, instead, it polymerizes into melanin, but that melanin is packaged up into granules which bind dopamine, I think, and stabilize it. And okay, so a lot of, okay, this is where I was like, holy crap. And again, here's that illustration of the pi conjugated system, and showing what if you excited an electron in there and you had this delocalized, essentially charge. That's really turns out to be really key to understanding why the pigment molecules form vesicles that effectively have a charged shell around them, which leads to a lot of the vesicle trapping ideas that I'm going to bring out ideally. So here are some of the questions I had when I was talking to Gary Miller's lab and asking for support. What do you think about these ideas? You've got credibility in the field. I don't. What had happened was I was trying to publish our finding that nicotine induces a fluorescent signal in the presence of melanin. And why was that important? Nicotine is protective against Parkinson's disease. I know a whole other story I haven't even introduced yet. You're going, what? Yeah, nicotine. Smokers have a lower incidence of Parkinson's disease. And one of the first things I thought was, I'd be willing to bet that nicotine interacts with melanin in the brain and essentially prevents things from binding that are more toxic. And that's sort of counterintuitive. It's like a prophylactic binding. Or if there's a reversible binding to that thing, then maybe things like MPP plus would have a harder time getting in there and causing trouble. That was my thinking. So this fact that they bound chemicals. So here's what I wanted to know: is toxin binding to neuromelanin a general mechanism of Parkinsonian inducing agents? Is there an endogenous substrate for neuromelanin? What is it? Is it dopamine, in fact? It sure seems like it fits the bill. If MPP plus is taken up into dopaminergic neurons because it resembles dopamine and is taken up through dopaminergic transporters, why is nobody asking the question whether dopamine binds to melanin in the brain? This would seem like such an obvious question to me, and yet it hadn't been asked. The functional, magnetic, and optical properties, this is where Peter Forehand comes in, and suddenly the dam is burst, and we've had some breakthroughs that But again, this is really just back to the pigment, the neuromelanin, what led me to these ideas. Here was another key finding. I gave a presentation in West Virginia University to the neuroscience group. There was a big group that met on a weekly basis or monthly or something like that and gave presentations in a big seminar hall. All the neurophysicians would come there. And this, I gave a presentation one time, and this paper was something I showed that it's related to the concept that some of you might have heard of, that neurons die over your life. So, yeah, only the weak ones. Yeah, but you know, you've heard jokes like that. Anyway, in other words, if you took a tissue slice and you imagine finding a neuron in it, a synapse, and what would you do to look for, is it a dopaminergic neuron? Well, what you actually do, it turns out, if you have a slide, you got a tissue slice, and you look at it under the microscope, here's your slide, and what you do is you stain. And in this case, they stain with an antibody, anti-tyrosine hydroxylase. Okay, and so back to that amino acid tyrosine. I started by telling you a story about how this amino acid spontaneously loses its carboxyl group, and that thing is known as tyramine, and that people that eat a lot of tyramine in their diet are sometimes subject to something called the cheese effect if they're taking MAOB inhibitors. In other words, you can take tyramine and you can add a hydroxyl group to it and you get dopamine. But you can also take tyrosine and add a hydroxyl group to it and then remove the carboxyl group and you still get dopamine. And that's what happens in the normal dopaminergic neuron. And so tyrosine hydroxylase is the enzyme associated with the production of tyrosine in a young dopaminergic neuron. And so how do you look for dopaminergic neurons in the tissue? Well, you look with anti-tyrosine hydroxylase. And so what people thought for a long time was that these neurons were dying, that they were disappearing as an animal got older. And they said that because they didn't stain with tyrosine hydroxylase antibodies anymore. In other words, the protein, the enzyme, the thing that would if you got to go understand antibodies, and I'm not going to do that in this lecture, but antibodies, it only shows that there's protein there or not. It doesn't really tell you much about what's actually happening in the cell. It doesn't tell you anything about whether that cell is actually a dopaminergic neuron or not. In other words, if dopamine was made or taken up or somehow stored in those cells, not through the enzyme tyrosine hydroxylase, you wouldn't see the enzyme anymore, but it could still well be a dopaminergic neuron. And that's what this paper showed in squirrel monkeys, where it looked at young, middle-aged, and old squirrel monkeys, and it counted neurons that had melanin in them versus ones that stained with tyrosine hydroxylase. And so that's shown here in some neurons that only stained with tyrosine hydroxylase. They look like in this one in A. And in C, by contrast, this is what neuromelanin granules look like in those cells. They're very black, compartmentalized into lipid vesicles. And here is a cell that has both tyrosine hydroxylase and neuromelanin in it, in panel B here. And this study counted those neurons in young, middle-aged, and old animals, squirrel monkeys. And it showed that the total number, if you include both of those, whether it had granules or stained with tyrosine hydroxylase, did not change over the lifetime of those animals. But the proportion that had tyrosine hydroxylase only staining. In other words, presumably they were making dopamine from the amino acid tyrosine. That ability decreased in older animals. And what happened at the same time, apparently, those cells had an increase in melanin. That is, the proportion of cells that were melanin only increased drastically in the older animals. The proportion that were expressing tyrosine hydroxylase goes down almost to zero in the old animals. And so you tell me, if this is a dopaminergic neuron, and it is, because it's in the substantia nigra, which is responsible for the release of dopamine to the striatum and all the other pathways, how does it make dopamine? How does it get dopamine if it doesn't make it through what's considered the synthetic pathway? This was my big question. Does it simply rely on the granule of melanin itself acting as a sort of magnet to draw in things that are pi conjugated systems? This is more on that same paper. That is, oh, wait a minute. Yes. In addition, this was the data that convinced me that, yes, you can kill any neuron if you add MPTP to them, because it's taken up into dopaminergic neurons and it disrupts mitochondrial electron transport. But if you actually look at what happens in an animal when you give them MPTP, the pigmented neurons were the ones that were particularly susceptible. So I'm trying to go through all that data right now. I'm getting kind of tired of talking, but we can come back to that sometime. Look up that paper. Pigmented cells were particularly susceptible to MPTP toxicity. That's a key finding. And so here's more thinking. I'll try and skip through the rest of this because we've got the gist of it, of the ideas, and the rest of this talk is sort of what happened between then and now, and a lot of it is repetitive, and I've sort of introduced the basics of the idea. There may be a couple more things in here that I'll show you just for interest. But this was part of my thinking in terms of why did monoamines go into neurons in the first place? Why do things partition where they do? What is it about neuronal signaling and voltage? And here is more on thinking about what happens around pigment molecules. That is, they're redox active. What does that mean? They continue potentially to release electrons, and this ties in with something that, oh crap, I don't think I can explain that very well on this lecture. At least I don't want to take the time to, because this one's already getting out of hand. And I'll be doing more, I think, over time, particularly if this video gets any sort of reception at all. So let me sort of skip through here. Again, more, as I started scanning the literature and thinking about where these ideas, possible, and I kept finding more and more stuff that said, oh my God, look at the properties of this thing. You can't tell me this thing is not functional in the brain. I just couldn't. It was just mind-boggling to me. I went to a neuroscience meeting, thousands of posters, none of them focusing on melanins. None of them. It boggled my mind. I talked to a guy from the Michael J. Fox Foundation. I said, what's neuromelanin doing in the brain? He said, neuromelanin, that's garbage, isn't it? And I kind of rolled my eyes. I talked to a guy that was in West Virginia University who came and gave a lecture. He was supposed to be the most famous eye person in the world. I asked him, what's melanin doing in the back of the eye? He said, melanin, that's a protein, isn't it? And I rolled my eyes again. When I had my ideas, I called up Luigi Zecca, whose name was all over those papers. And I said, Luigi, what do you think about melanin acting as a memory process to bind, store, and release dopamine? And he said, yeah, that's an interesting idea, but you should probably cool your heels a little bit because, and he came and gave a lecture at West Virginia. I invited him. He stayed at my house. We were walking down the hall and I said, Dr. Zecca, have you had trouble getting your colleagues to appreciate the significance of this melanin polymer? And he sighed and he looked at me and he said, I spent 20 years convincing my colleagues that neuromelanin binds iron. So that was when I went, oh, now I think I understand what I'm up against when you think about the dogma in the literature. And he wasn't entirely on board either. He liked my ideas, but he was still too ingrained in the, yeah, but nicotine binds to the receptor here, and so it can't be protective by binding to neuromelanin because we already know it acts through the nicotinic acetylcholine receptor. But I say bullshit. Nicotine goes right through the skin, and it is certainly not limited in action to bind to some receptor on the surface of a cell the way it's pictured and described in the literature. So that's sort of another, side story. I got fired from West Virginia University, you guys. I'd had all these ideas. I'd sort of discovered the root cause of Parkinson's disease as far as I was concerned. This was an idea that needed to be shared with the world, and I'd spent all this time, my career, preparing for this moment. And I had been hired under the conditions that nothing specified that I needed grant money when I went up for promotion. But they changed the rules in the middle in West Virginia. And so it was an open question up until the day I walked into the dean's office in School of Medicine, and they handed me the letter from the dean of the School of Medicine saying I was not promoted to associate professor, and I was denied tenure, and I was given a 1-year terminal contract. I was mad at the world, you guys. I was really mad at the world. And I had quit drinking, and I was sort of about to go through a divorce. This is what came out. The long story. I started etching mirrors. I started trying to sell them. I ended up running into a woman that had a picture framing shop who was retiring. I said, fuck science. I'm going to go do art for a few years. And I had no training in art and no training in business. So this was sort of not really the smartest thing I've ever done, but it also sort of helped me get my head screwed on, right, for a few years. And I had a lot of really fun times in Morgantown working as an artist and meeting a lot of people that I never met when I was working at the university. Well, 3 years of doing this stuff, and my youngest daughter was going to leave with her mother to another state, and there was not really much reason for me to stay in Morgantown, and I wanted dental insurance. I wanted to be able to support Jenica when she got to college, and so I accepted the job at West Virginia University, and we started playing around with things that we could do there. We could grow neurons in culture. We could add L-DOPA to them, and we could sort of mimic the experiments that Salter was doing in a way. We could show that if you add L-DOPA, you pigmented those neurons. And so we were interested in the idea that these pigmented neurons were going to be more susceptible to MPTP toxicity, and we were going to show that in cell culture. and add to that literature. And we also showed that you can add the precursor of 6-hydroxydopamine and effectively also cause melanin production. We never published this, but we probably should have. It didn't quite fit into anything at the time. When you looked at a combination of adding L-dopa, a precursor to dopamine, and the precursor to 6-hydroxydopamine, they also pigment the neurons. That's what this is showing. Essentially, here's neurons in the upper left in culture, and then after we add it over time, they blacken because they take it up and they polymerize the stuff and they form those granules. And we were interested in seeing if we could show the downregulation of that enzyme tyrosine hydroxylase in these cultured neurons. And so that's what this was, that you could actually buy an anti-tyrosine hydroxylase antibody, but you could also buy an anti-dopamine antibody at this point, which wasn't available years ago. And so we could sort of see in these cultured neurons where the cells were active, whether they had a nucleus, that's what the DAPI staining does, whether they had dopamine in them, and whether that dopamine was localized or diffuse. We could sort of see that in some of our cell culture. And we were looking for the presence of tyrosine hydroxylase. And we never did quite optimize these experiments. So again, this is unpublished preliminary data done with undergraduates that wasn't repeated enough and didn't give clean enough data to publish it. But it was sure shit interesting and was on the right track. So a couple other things. One of the next things I did, I had a class, an upper level integrated laboratory it was called, and I had 8 students, and I said, well, I'm going to put them to work doing neuromelanin studies. And we bought ourselves a new instrument, a little benchtop fluorescent spectrophotometer, which opened up a lot of doors. It allowed me to think about doing fluorescent spectroscopy in very small volumes with small amounts of material, such as a synthetic melanin analog. And so what we did in that laboratory was I had a student make a recipe that Dr. Zeka's laboratory had published. This is shown here, where we mixed dopamine, added some cysteine. Oh, that's not this recipe, sorry. These were some earlier studies, forgive me, where we hadn't quite understood the difference between iron-bound melanin and iron-free melanin. But we were looking for, and forgive me, this was before we got in the fluorescent spectrophotometer. My thought was that if Parkinson's disease is essentially caused by things that bind to melanin, one of the things that I could do at a small undergraduate institution with undergraduates was try and do some spectroscopy to show what things interacted with melanin and how do you show things interact with the chemical. This is often where spectroscopy comes in, where you shine a light through something, you see how the light is absorbed, and you see how that changes with concentrations or alterations in the reaction, things that are going on in there. So without going into any detail really here, we had some hints that dopamine polymers, that is a neuromelanin bound 6-hydroxydopamine. And shoot, the axes got messed up in here, and so you can't tell what any of this data means. Here's another diagram essentially showing, I know the label is gone on this one too. This was showing the concentrations of these things. Basically, the short version was we couldn't tell very much from visible spectroscopy. We had some hints that these things were interacting. We saw some peaks, which is sort of a sign that as you increase the concentration of something, there's an inflection point. It's not a linear relationship with something was interacting with the melanin that was altering its absorbance. But it's hard to measure because melanin is black. What does that imply? It implies it absorbs all of the colors of the rainbow. So how are you going to shine a light through it and get any information out of that? You can't. You can't really. Not very useful information. We tried, but we didn't get much. And that's again where the fluorescence spectrophotometer comes in. That is, fluorescence is a whole different ball of wax. That it often gives you a much more sensitive signal. And so I said to one of my students in integrated lab, I said, why don't we'll make some melanin, neuromelanin analog, and toss in some nicotine, because we know nicotine protects against Parkinson's disease. I hypothesize that part of that protection is binding to neuromelanin, and if so, we should see a signal. We should see something that changes when neuromelanin is in the presence of nicotine. And one of my students showed this, that nicotine and neuromelanin fluoresce, and there is a saturable endpoint of that, which says there is a specific binding interaction between neuromelanin and nicotine. Okay, well, so what? That doesn't prove it has anything to do with Parkinson's or anything about protection from Parkinson's. How could I use that to my advantage and publish something that would give me credibility in the field of neuromelanin? What I figured was that this gave me access to my question of, how do I tell what things are potentially going to cause Parkinson's disease? That is, what things are going to bind to neuromelanin? How would I know if I couldn't measure them through UV visible spectrophotometry and they didn't have a fluorescence signal? What I knew was that they would almost certainly disrupt the fluorescence signal of nicotine. And so this is what we published. This is how I got credibility as a scientist in neuromelanin and in neurochemistry, because I'm cytochrome P450s, pharmacogenetics. And I said, this is potentially an assay. And one of the things we showed was that if you try and compete with nicotine, dopamine competes with that signal and actually increases it at very low concentrations. And so you get an inflection point at very low concentrations and then a second one at higher concentrations. This peak right here says to me dopamine is binding to neuromelanin at very low concentrations, micromolar concentrations, submillimolar concentrations. Dopamine is probably the natural substrate that binds to melanin in the brain. And almost certainly it's involved in re-releasing that dopamine. And if it can't do that, you've got Parkinson's disease. So this was my conclusion. And we published that, and I hypothesized that this was going to occur. And so I wrote up the hypothesis paper, which is neuromelanin is not a spectator. And I think with that, I'm going to stop this recording. My voice is about had it for the night, and we'll see if I can upload this and see if anybody ever watches it. But I hope you enjoyed it. If you do watch it, and I hope it makes you start thinking about things in a new light.